Hi, so we're continuing with my notes for life science and cell biology in a nutshell. We're learning about the cell membrane. So, the cell membrane, which is also called the plasma membrane or plasma, is the semi permeable lipid bilayer common to all living cells. It contains a variety of biological molecules we mentioned before. Primarily, these are proteins and lipids, as well as uh, carbohydrates, which are bound to these proteins and lipids. Um, so these molecules are involved in a vast array of cellular processes having to do with the membrane and various reactions within the cell. The membrane serves as an attachment point for both the intracellular cytoskeleton inside the cell and also, if present, the cell wall for plants and bacteria cells. So, its cell membrane is important. Phospholipids have been widely used um, um, also in experiments to prepare liposomal, um, other nano formulations of topical, oral, and parenteral drugs for different reasons like improved bioavailability, reduced toxicity, increased permeability across membranes. So these can be synthesized for enhancing the function of cells. Liposomes are often composed of phosphatidylcholine enriched phospholipids and may also contain mixed phospholipid chains with surfactant properties. So, surface properties. The ethosomal formulation of ketoconazole using phospholipids is a promising option for transdermal delivery in fungal infections. So, these are some examples. Um, Phospholipids are optically highly um, birefringent, which means their refractive index is different along their axis as opposed to perpendicular. Measurement of birefringence can be achieved using cross polarizers in a microscope to obtain an image of, for example, vesicle walls, or using techniques such as dual polarization, interferometry, interferometry to quantify lipid order or the disruption in the supported bilayers. So there's various ways of figuring out the makeup of layers. Analysis. There are no simple methods available for analysis of phospholipids since the close range of polarity between different phospholipid species makes detection difficult. Oil chemists often use spectroscopy to determine total phosphorus abundance and calculate approximate mass of phospholipids based on molecular weight of expected fatty acid species. Modern lipid profiling employs more absolute methods of analysis with nuclear magnetic resonance spectroscopy (NMR), particularly called 31P NMR, while HPLC-LSD provides relative values. Phospholipid synthesis occurs in the cytosol adjacent to the endoplasmic reticulum membrane that is studied with proteins that act in synthesis. For example, GPAT and LPAT acyltransferases, phosphatase, choline phosphotransferase, Um, and allocation called flipase and flopase. So these are different proteins um, involved in phospholipid production. Eventually, a vesicle will bud off from the ER containing phospholipid uh, molecules, which are destined for the cytoplasmic cellular membrane on its exterior leaflet, and as well destined for the exoplasmic cellular membrane in the inner leaflet. So either for the outside or the inside of the bilayer. In signal transduction, some types of phospholipid can be split to produce products that function as second messengers in signal transduction. Examples include phosphatidylinositol, 4,5-bisphosphate, PIP2, that can be split by the enzyme phospholipase C into inositol triphosphate, IP3, and diacylglycerol, DAG, which both carry out the functions of the GQ type of G protein and responds to various stimuli and intervene in various processes, from long-term depression in neurons to leukocyte signal pathways started by chemokine receptors. Phospholipids also intervene in prostaglandin signal pathways as the raw material used by lipase enzymes to produce the prostaglandin precursors. In plants, they serve as the raw material to produce jasmonic acid, a plant hormone similar in structure to prostaglandins that mediates defensive responses against pathogens. So next section is food technology. Phospholipids can act as emulsifiers, enabling oils to form a colloid with water. Phospholipids are one of the components of lecithin, which is found in egg yolks, as well as being extracted from soybeans and is used as a food additive in many products, can be purchased as a dietary supplement. Lysolecithins are typically used for water oil emulsions like margarine due to their higher HLB ratio. Um, okay, we have a diagram of cholesterol showing the hydroxyl group making the head polar and hydrophilic, attracted to the phosphate heads on the periphery of the membrane, which are the phospholipid molecules. Carbon rings. Cholesterol is not classed as a fat or an oil, it is a steroid. And then the nonpolar hydrophobic tail is attracted to the hydrophobic tail of phospholipids in the center of the membrane, the bilayer. So it's showing the two layers of the phospholipids facing one another and the cholesterol molecules that interact with it. Cholesterol is a bipolar molecule, just like phospholipids, both hydrophilic due to the polar hydroxy groups and hydrophobic due to the hydrocarbon rings and the hydrocarbon chains. It serves to increase stability due to its polar aspect, as well as to increase fluidity of the layer due to its non-polar aspect. So its functional advantage depends on the cell's environment. So stability, because two phospholipids between the phospholipids, it reduces the lateral movement of the molecules, causing more integrity of the membrane bilayer, helping control permeability of the membrane to molecules as well. So the interaction makes the membrane more stable.